Hi everyone, it's Mr. Sinti. Today I'd like to talk to you about muscle fiber types. That's right. Muscle tissue is a group of cells collectively that helps us move. And when you consider what a muscle fiber is also known as a muscle cell. And so today's conversation is going to sort of circle around muscle fibers and how they contract and how there's different types of muscle fibers and some various aspects of energy production. But I want to start off a little bit with a review. You may know this already, but just in case, we'll sort of ease into the conversation if you don't mind. Muscle itself can be primarily divided into three basic types. There's skeletal muscle, which is what this conversation is centered around, which is attached to skeleton, helps us move. But there's also, and I wanted to identify, cardiac muscle, which is found only in the heart, and smooth muscle, which is found really ubiquitously around the body, but mainly that lines the alimentary canal, the digestive tract, and also that makes up the lining of our blood vessels. And so the function of skeletal muscles, in addition to them being attached to the skeletal system to help us move, they help us to maintain posture and body position uh, in terms of muscle tone. That'll come up in the conversation. They also help to maintain body temperature. There's a potpourri of functions for muscles in addition to moving. Now, body temperature may seem obvious because the repeated contraction of muscle generates a lot of friction and heat, and so that helps to maintain body temperature. This one is a little bit, this final function is a little bit more obscure, and I find it interesting. It serves as a nutrient reserve. What does that mean? That means, in addition to the muscle fiber or muscle cell storing a polysaccharide glycogen, under times of distress and deprivation, the proteins found in muscle can actually be broken down as a source of energy, and they can also be converted. Those amino acids could be converted into glucose, and so it is a reserve for nutrients. Let's talk a little bit about the hierarchy of a muscle, to sort of the basic organization of a muscle cell, so that we can get into a conversation about muscle fiber type. So if you look at a muscle, a typical muscle, you'll notice that the muscle itself is surrounded by connective tissue and something called epi, meaning upon, epimysium. And so right here, the whole muscle surrounding, and each of these individual units inside the big muscle are known as fascicles. And so muscle is composed of multiple fascicles. Okay? And when you look at a fascicle, if you were to break that down and pull out a single fascicle, you'll notice that the fascicle itself is surrounded by connective tissue known as perimysium, or again, around the muscle, mesium meaning muscle. Inside a fascicle, there's also bundles of muscle fibers, and these muscle fibers are known as the muscle cells, and they're rather long, if you will. This is a muscle fiber being pulled out of a fascicle right there. So let's take a look at this. Here's a muscle fiber or cell, and one of the things that might strike your attention is like, whoa, what is going on with all of this red in here? These are all myofibrils, in other words, sheets and sheets of protein that are found inside muscle cells that are very important for muscle contraction. Okay, and so the membrane of a muscle fiber is known as sarcoplasma. Okay, so the membrane of the cell. All right, so we're going to get into that. I'm sorry, the sarcolemma is the membrane of the cell. Sarcoplasm is the fluid of the cell. I misspoke there. Sorry about that. So when you look at a muscle fiber, there's some uniqueness to this. That first off, they're very long cells, and that's one characteristic. They have all of these protein myofibrils inside, and they're multinucleated. Multinucleated. So when you look at this is a look at under the light microscope. You can actually see these nuclei. And so how did that come into being? Well, a muscle fiber originated from these myoblast cells. And when they fuse together, these are sort of ancestral cells. And when they fuse together, you can see how there's more than one nucleus available in a muscle fiber. But one of its main functions is the fact that the cell is so long it needs to have that kind of control in terms of making protein. You may know that what the nucleus functions in is that it houses DNA, and therefore it codes for protein. And so it's useful to have all those nuclei in the cell. And this is a really cool close-up under the light microscope. Do you notice these lines that are going across right here in a muscle fiber? This is known as striations. And sometimes skeletal muscle, you may be familiar with this, is also referred to as striated muscle because of that. And what those striations are the sort of dark light that you're seeing when you look at the myofibrils in protein. And let me make that a little bit more clear. When you look at a single muscle fiber or a muscle cell, and you were to pull out some of those myofibrils, you're going to notice that the proteins themselves, where I'm pointing with the cursor right there, are made up of different kinds of proteins, and they overlap with one another, creating dark regions and light regions, which account for the striations. And again, here's the mitochondria that are found inside the muscle cell, which produce ATP. And here are the proteins, and here is the liquid, the sarcoplasma, and here's the sarcolemma, which is the membrane. Okay. Now this is a closer look even still, and you can see inside the one muscle cell, you can see all the myofibrils right here, and you can see how in some regions there's thick and thin filaments overlapping, which gives the dark striation, dark light. Okay, and what you're seeing here in blue is the endoplasmic reticulum, which is being emphasized. The endoplasmic reticulum in a muscle fiber is actually called sarcoplasmic reticulum because it's the endoplasmic reticulum of the muscle. And so we're going to find out, if you are familiar with this already, this is a reservoir of calcium ions, which are very important in regulating muscle contraction. They initiate muscle contraction. Very important, calcium in muscle fibers. And here you can see the myofibril being pulled out, and you can see thick filaments, which are made up of a protein called myosin, and the thin filament, which is made up of a very popular protein called actin. And collectively, those slide past each other, and in so doing, when the thick and thin filaments slide past each other, it actually causes the myofibrils to shorten and therefore contract, and therefore the whole muscle fiber contracts, therefore the whole fascicle contracts, and therefore the whole muscle contracts. So this is a cool look right here, like a little bit of a cross section. You can see the myofibrils, the mitochondria, which is producing ATP. And again, just another perspective of the muscle fiber, so you can get a really good sense of it. You'll notice that there inside the muscle cell, there's these columns, if you will, called T-tubes, which will help to assist the action potential, which is coming from a motor neuron, to be able to reach all aspects of the muscle fiber since it's so long. And so that helps to conduct the action potential. That's what that is. And then finally, this picture where we're just looking at the protein. And you can see here's the sarcoplasmic reticulum, which is a storage of calcium right over here. And one of the things that I wanted to emphasize when I was saying that the muscle fiber shortens, the way we define that in anatomy and physiology is we say that there's units inside the muscle fiber, and these units are called sarcomeres. Mer meaning part, like monomer, polymer. These are, again, sarco meaning flesh, so fleshmer. It's kind of a weird thing. So from Z line to Z line, which is right here, and right here, defines one sarcomere. Here's a better picture of this. And so a sarcomere is the unit of a muscle fiber. This is what is shortening. Each sarcomere is shortening. And it goes from Z line to Z line. And you can see the reason that you could see this is that this is again, a zigzag or Z line, a zigzag column of protein made up of the protein made up of the protein actin. This is a thin filament. This thick purple protein is myosin. Notice how it's really thick right there, and it has these little sort of arms extending from it called cross bridges. And those are going to attach to the thin filaments and cause the sarcomere to sort of come across each other and shorten. And notice where thick and thin filaments overlap right in here, this is referred to as the A band. And notice how it's a little dark right there. And then there's a lighter region right here where there's no thin filaments. And again, you can keep going with this. You can say that, all right, the H band is right in here. It's kind of light. The M line or middle line is right in the center, as you can see right over here. Okay, so Z line to Z line makes up a sarcomere. Now, backing away just a moment from this, when you actually look at these myofibrils with inside one of the muscle cells, nerves actually control, because skeletal muscle is voluntary. So there are axons that are reaching each of the myofibrils. And I'm sorry, the yes, reaching the muscle cell, and where they come together, where the axon terminal reaches the muscle cell, is known as the neuromuscular junction, where those two things come together. This is a really nice picture, I think, of the axon of a motor neuron. And you can see here's the axon of a motor neuron. Down here in red is the muscle cell itself. And then here are the axons. They're sort of like little pads right there. And what's fascinating here is that inside the motor neuron, so inside the neuron, there are vesicles containing chemicals called neurotransmitter. And then those are released through exocytosis into this synaptic cleft. In other words, this area right in here, which is the junction between a neuron and a muscle fiber or muscle cell. And the chemical that is released is going to stimulate the muscle to contract, and that molecule is known as acetylcholine, and it's a neurotransmitter, acetylcholine. So it's really cool when an action potential comes down. This video isn't about that, but I do have other videos on action potentials. If you're interested, I welcome you to check that out. It's pretty interesting. So the action potential comes down, the acetylcholine is released right in here, and what happens is it's picked up by a protein receptor in the muscle cell membrane, the sarcolemma. Okay, and what happens, as you can see here, you'll notice that the membrane of the muscle cell is convoluted, so there's more surface area and more ability to receive the acetylcholine. Okay, and so there's folds of the sarcolemma at the motor end plate of the motor neuron. And so these vesicles containing acetylcholine release this, and they go into this space right here. And what happens is they're picked up by the acetylcholine receptor. And this is a chemical gated ion channel. Now, I don't want to assume total understanding of that. I do have a video that discusses that as well. Sorry to deflect it, but I'll briefly discuss it. This video isn't about that in particular, but I feel like it's necessary to sort of attend to it. Acetylcholine comes, and when it binds to the protein, it causes the protein to change shape temporarily, so it will allow sodium to actually pass. Sodium's an ion that is impermeable in the phospholipid bilayer, but once acetylcholine attaches to it, sodium can come into the cell. Now, why it comes into the cell is because there's a tremendous amount of sodium outside of the cell, so it's in high concentration. Check this out. When sodium comes into the muscle cell, this is the muscle cell down below. When sodium comes in, it's going to make the sodium, because it's positively charged, it's going to make the muscle cell become more and more positively charged. And that's going to cause the muscle cell to be able to contract ultimately. Because why is that? Because this is sort of a diagram showing like each of the steps involved. So acetylcholine is released by the neuron. It's picked up in the neuromuscular junction by acetylcholine receptors. That causes an action potential, which is again a change in charge from the resting potential of a nerve cell, of a muscle cell, which is negative, and as it becomes more positive, that transmits down these T tubes, And what ultimately it reaches the T tube and the action potential is generated, and then it reaches the sarcoplasmic reticulum. Now, this is important. The action potential causes the sarcoplasmic reticulum to release calcium ions. And calcium ions are regulatory in terms of they can actually cause muscle contraction to occur. And what they do is they make actin available to be attached to myosin, which then causes the sarcomere to shorten. Now, I will come back to this diagram, but I need a close-up of what I'm trying to describe in terms of calcium regulation. This is actin, these golden molecules, and they're polymers, and they're made up of two strands. But there are places where the myosin cross-bridge attaches, and those are known as the cross-bridge binding sites, right over here. But those are blocked by a regulatory protein called tropomyosin. So it's blocked. So the thick filament cannot grab the thin filament because it's being blocked by tropomyosin. And attached to tropomyosin is another protein called troponin. It's made up of a tetramer. It's this little protein right here, and it's blocking tropomyosin. And so what happens is calcium that's released from the sarcoplasmic reticulum of the muscle fiber attaches to the protein troponin, which causes it to change shape, which then causes tropomyosin to change shape, which then causes, they sort of look like little olives to me, which allow these active sites, which are the myosin binding sites, to become available. And then what happens, let me go back here, the myosin cross bridge is now able to attach to the actin molecule and cause it to be pulled toward the M line. In other words, the Z line and Z line of one sarcomere come past each other, which is known as the sliding filament theory. And that is contraction, when the sarcomere shortens. How about that? Pretty cool. All right, and so then what happens, what stops muscle contraction? Well, ultimately what's happening is the acetylcholine, it could simply diffuse out of this particular area, but there's also a special enzyme that needs to break it down quickly so that we have control. Contract the muscle, don't contract the muscle. Contract the muscle, don't contract the muscle. So it's acetylcholine esterase. How's that? Breaks down the acetylcholine, and so therefore no more action potential. And so what happens is, do you notice here in the sarcoplasmic reticulum, this may seem like a detail, but in my judgment, it's not. It's rather important. So if you can take note of this. Do you notice the concentration of calcium inside the sarcoplasmic reticulum is very high? And of course, when the action potential came, we just talked about how calcium is released in the cytoplasm. But then it has to be actively pumped back into the sarcoplasmic reticulum because it's against the concentration gradient. So that requires ATP to pull calcium back out and stop contraction. And without calcium, what's happening? Whoops, let me go back here. Without calcium, what's going to occur is troponin is then going to return to its normal conformation, which is then going to have tropomyosin block the binding sites, and contraction will be ceased. Okay, and so notice here there's going to be no active sites are covered, and so therefore the myosin isn't able to pull on the actin, and therefore contraction ends and the muscle relaxation occurs. But it's an active process, if you will, because it does require ATP to pump the calcium ions back into the sarcoplasmic reticulum. So concentration duration depends on, and this may be obvious, but I'll state it anyway. It's the duration of the neuronal stimulus affects concentration duration, and it also the amount of calcium ions in the sarcoplasm affects it. So if you had repeated stimuli and you had just a ton of calcium inside the cytoplasm of the muscle cell, you're going to get in more intense concentration. And also the availability of ATP. I didn't mention it per se. I mentioned how ATP is necessary to actively pump calcium back into the sarcoplasmic reticulum. But ATP is also very important in attaching to the myosin cross bridge to disengage the myosin cross bridge and cause it to change shape and if you will, with lacking a better analogy, to sort of cause it to cock back like a revolver cock would pull back. So it allows the myosin cross bridge to free itself up and be prepared for another power stroke, as it's referred. So relaxation, just to review this, calcium concentrations would fall in the cell. Calcium then detaches from troponin, and therefore the active sites are covered back up again, and therefore myosin crossbridge wouldn't be able to attach to the active site. Now, what I find particularly interesting, and it's not necessarily a pleasant conversation, but I find it kind of interesting. I hope you uh, enjoy this, and I think you might be aware of the fact that when someone passes away and dies, they're not breathing anymore, they're not producing any more energy, they're not eating, and so ultimately, without oxygen, the muscle cells are not going to be able to produce ATP. And what happens is that the body becomes very stiff. It's known as rigor mortis. And so sometimes the body is actually referred to as a stiff. And forensic scientists, and again, I'm not a forensic scientist, but forensic scientists are able to take a look at the degree of rigor mortis and to know, and they can determine the time of death. It's quite amazing. I welcome you to sort of look into that if you're fascinated by this. But let me discuss this as it relates to what we've been talking about in terms of muscle contraction and relaxation. Ions are pumped in order to cease the function, and when calcium ions are not able to go back into the sarcoplasmic reticulum, they're not able to go back because the cells are running out of ATP because the person has died. Without ATP, what will ultimately happen is calcium will eventually just leak out of the sarcoplasmic reticulum. And that's what this picture is right here. This is actually a transmission, electron micrograph of the sarcoplasmic reticulum of a sarcomer. And what's happening is when the calcium is coming out, what it's going to do is it's going to attach to the troponin, if you recall that, and therefore it's going to cause the muscle fibers for myosin crossbridge to attach, and it's going to cause prolonged contraction, sustained contraction, and so therefore all the muscle in a dead person's body are going to be contracting, causing stiffness to occur. It's remarkable. And then also, I might add this, without ATP, the myosin heads don't detach, and so they're permanently locked into place. Because if you recall, in order for the power stroke to occur, in order for myosin, when it pulls, another ATP needs to attach to the cross bridge in order for it to detach, in order for it to go back and pull, back and pull. And so it's a combination of those two events where the muscles are continuously contracting, which causes this. Now, this part of the video is a discussion. We're getting into the fact into these muscle fiber types, and muscle fiber types have a lot to do with this concept, which is the frequency of stimulation. So when a single neuron stimulation produces a contraction, it's referred to as a twitch. You may be familiar with that term. And it's very fast, but there's a range from 7 to 100 milliseconds. And so there's a range here in terms of a twitch or a single contraction. Now, in order to have sustained muscle contraction, if you're running, for example, it's going to require repeated stimuli or multiple twitches. But let me sort of just take a look, show you this diagram here in terms of a twitch, which I'm talking about a contraction, but it's not just a contraction. There's 3 phases to this, or 3 periods to a twitch, which is an increase in muscle tension over time. All right, that's what this is, an increase of muscle tension over time. And so there's a latent period because it takes a little time for the action potential to move through the membrane or sarcolemma of the muscle fiber, releasing the calcium from the sarcoplasmic reticulum. So there's a little bit of a lag period right in here in the beginning. But once calcium's released, make no mistake, it's going to bind troponin, therefore move tropomyosin, and then the myosin cross bridge is going to attach to actin and pull it in. And so that's what's the contraction phase, or the power stroke, if you will. Calcium ions are going to bind, and then the tension reaches a peak. Okay, and then there's relaxation phase, and that may be obvious. ATP is required to pull calcium back into the sarcoplasmic reticulum. Therefore, the active sites are covered up again, and the tension falls to a resting level. So there's a twitch right there over time. And so, in terms of milliseconds, there's range on this. And then here's the peak, maximum tension development. So there's a latent period, a contraction phase, and then a relaxation phase. Okay? Now, let me go back to this. Now, if you were to study this further, you could notice there can be multiple twitches occurring. And you can really get into that. And what's fascinating is when you have multiple stimulation occurring simultaneously, what can happen is that these peaks will actually increase, and it's sort of like a staircase. It causes more and more calcium to be present, and therefore the tension strength increases. It's kind of interesting. So I wanted to bring your attention to this chemical called ATP. I've been talking about it, but just in case, to review the structure of this, it is made up of a ribose sugar and 3 phosphates and an adenine nitrogenous base, which is a purine. This is a monomer. If you're familiar with genetics, this is the monomer of RNA. So it's adenosine triphosphate. And the cell uses this for energy because these 3 phosphate groups, which are negatively charged, they really don't like being next to each other. And that's a simplification. But if water were to cleave the 3rd phosphate, you would have adenosine diphosphate, or ADP. This is ATP for triphosphate, 3 of them. And what the cell can do cleverly is when it removes that phosphate from ATP, it could attach it or stick it onto another protein. And when a protein receives a negative phosphate group, it'll change shape. And when it changes shape, it allows it to move, and therefore work can be done. And that is what is happening. That is what, when you stick a phosphate onto the myosin cross bridge, it's going to cause it to release from the actin, and it's going to cause it to pull back. It's pretty awesome. So sustain, as this again, sorry for being obvious, but sustained muscle contraction, again, like walking or swimming, requires a lot of ATP. And so muscles store enough energy to start the contraction. So there's energy, ATP, present in the cytoplasm of the muscle cell. But as soon as you start to contract those muscles repetitively, you're going to need to manufacture ATP as needed. And so this is going to be where our conversation is going now, which is not only do you need to make ATP, and there's a couple ways to make it, which we'll talk about, but you also need sources of phosphate. Now, phosphates can be floating. This particular video isn't a biochemistry video, but I need to talk about it just for a second. The phosphates inorganic phosphates are floating around in the cytoplasm, but phosphates could also be attached to organic, meaning carbon-containing molecules. And muscle cells have a lot of this chemical called creatine phosphate. Here's the phosphate functional group, or CP for short. This is how muscle cells will store phosphates in order to add a phosphate onto ADP to make ATP. Now, in order to add it, it requires energy. This again may be obvious, but it requires the consumption of food and burning of food in order to make the energy necessary to create ATP, and then ATP then is used as a source of currency to cause the muscle to contract. And creatine phosphate is a source of phosphate to make all this happen. Now, this in terms of metabolism, ATP generation is a very large conversation. And so this is, I apologize for its brevity here, but let me, again, I don't want to particularly promote videos, or maybe I do, but I do have videos, if you want to look them up. I have a video on uh, the production of ATP in detail in a process known as glycolysis. I have a video on the citric acid cycle, sometimes known as the Krebs cycle. And I also have a separate video on what is known as the electron transport system, or oxidative with oxygen phosphorylation. In other words, the transfer of phosphate, what I was just talking about, onto ADP to make ATP. But I'll say this: in order to generate the energy necessary to make ATP, that energy comes primarily in our body from carbohydrates and fats. And fats are made up of a glycerol and 3 fatty acids, and they're known as triglycerides. Fats contain twice the number of calories, or twice the ability of making, or 3 times in some instances, to generate the amount of ATP necessary. For every one glucose, for every one triglyceride, you would need 3 glucose in order to produce that same amount of energy. It is like 9 kilocalories per mole. And so what is happening is carbohydrates could be used to produce energy, fats can be used, and if necessary, proteins can even be broken down into amino acids, and those could be used. But the body doesn't really want to use protein for ATP production, only in case of a crisis. It would rather use those amino acids to build protein. And so, in general, there's 2 ways for the cell to make ATP, and that is aerobically with oxygen and anaerobically without oxygen. So aerobic and anaerobic metabolism. And the way aerobic metabolism works with oxygen is that, as I was mentioning before, fatty acids could be used and glucose could be used. And what happens is, in the cell, fatty acids and glucose are broken down in a pathway called glycolysis to a 3-carbon molecule called pyruvate. And if oxygen is present, it will cause pyruvate to enter into the mitochondria, and where certain metabolic reactions take place in order to generate a large amount of ATP. So aerobic metabolism makes a tremendous amount of ATP because oxygen is important for doing that. That's the Krebs cycle and something called the electron transport chain. Now, if a cell, muscle cell since we're talking about, is in oxygen debit, or it's in distress and there's not a lot of oxygen, so no oxygen is present, the cell can still make ATP, but not very much. It can do glycolysis and one more reaction producing a molecule called lactate or lactic acid. And so more about this. So as I was mentioning before, aerobic metabolism makes a tremendous amount of ATP per glucose molecule. And so it's able to pull all the energy out of glucose to make 34 ATP. And it does that in the mitochondria and in the inner membrane of the mitochondria. Again, this looks detailed, but what will happen is there's a molecule called NADH, which is, if there's an acronym, that means that there's going to be something here. So nicotinamide adenine dinucleotide. How do you like that? So all the food that we eat are basically converted in the Krebs cycle to what is called a reduced coenzyme, NADH, and then the energy in that molecule is stripped in the membrane of the mitochondria in order to create a membrane potential in the mitochondria, which then causes ATP to be produced. And that's known as oxidative, because oxygen is involved in this process of the electron transport chain. Oxidative phosphorylation. In other words, the transferring of a phosphate to ADP to make ATP. So how's that for saying it very shortly? Glycolysis is the process of taking glucose and turning it into pyruvate. And as you notice, glucose, if you notice this, there's 6 carbons in glucose and only 3 in pyruvate. And so you actually make 2 pyruvates. So you split glucose into 2 pyruvates. And in so doing, it's 10 steps to do that. And in so doing, you can generate a little bit of ATP even without oxygen. Although what can be confusing is that glycolysis is also the beginning pathway of aerobic cellular respiration. But if you remember, there's a little fork in the road. Without oxygen, the cell could still make a little bit of ATP without that. And so what glycolysis involves is just the breakdown of glucose, because it literally means sugar splitting or glycolysis. But here's the drawback. When the cell is exerting itself and the body isn't able to provide enough oxygen for the muscle cell, the glucose can be broken down into pyruvate in glycolysis, and 2 ATP, as I mentioned before, are produced. But then, in order to regenerate, and this is complicated, in order to regenerate NAD plus, which is a compound needed in glycolysis, and a redox reaction needs to take place, and in so doing, it produces lactate. And one of the problems of that is that lactate can then dissociate and release hydrogen ions and make lactic acid. And when the muscle cells are producing a large amount of lactic acid, you may be familiar with this. This isn't so good, and the cell can't maintain it because the acidity will cause trouble. And for a variety of reasons, it'll cause the other proteins in the cell to break down, and it throws off the metabolic reactions when there's too much lactic acid present. However, during moderate exercise, in other words, walking or jogging or slow swimming or just sort of easy going, capillaries are able to provide the much needed oxygen to the cell. The cells are able to take that oxygen and allow either glucose that is converted into pyruvate to go into the mitochondria, or it's allowing fatty acids to go into the mitochondria to be broken down in aerobic respiration to make a tremendous amount of ATP, which is what the myofibrils need in order to contract. So this is moderate activity. Cells use aerobic metabolism in order to generate ATP. So when you're exercising under control, meaning you're breathing and everything's kind of going easy going, the cell's able to produce a lot more ATP, and it's able to burn glycogen, which is a polysaccharide, and it's also able to burn fat. Because you're not going to be able to burn fat unless you're breathing lots of oxygen. So fat burning isn't quick exercise. And so if during peak activity, if you're sprinting or if you're really racing up a steep hill, the cell's going to be in oxygen debt. And so in order to get through that, all it can do is use glycolysis. And so it takes some of its glycogen, makes it into glucose, and then uses glycolysis to produce pyruvate. This, again, does not go into the mitochondria, and therefore it gets converted to lactate or lactic acid, which, again, causes buildup, and it inhibits a lot of the functionality of the cell, and it leads to fatigue and pain and soreness. And ultimately, it would hurt so much that you would have to stop your exercise. And so this is kind of what muscle fatigue is all about. When the muscle can no longer perform the required activity, it becomes fatigued. And that can be caused by a depletion of metabolic reserves. In other words, it could be lacking the sources of of glucose, but it could also be lacking ATP, and also muscle fatigue is caused by low pH or lactic acid buildup. So you can recover from this. And so after muscle fatigue, there's some time that's needed for the muscle to sort of like calm down or recover. And so oxygen then becomes more available, and then ultimately the cell is able to resupply its stockpile of ATP. And I might add, mitochondrial activities resume, and so the cell sort of gets back going again. And then I would also say part of recovery is taking in some more food, which would then produce more ATP. And so in order to remove the lactic acid, this is the Cori cycle. It could be a video in and of itself, so I won't go too far into it, but ultimately the lactic acid needs to be removed, and that's actually removed when lactic acid diffuses out of the muscle into the bloodstream. The liver sort of converts lactate to back to pyruvate, and then also glucose is stored in the liver in the form of glycogen. So glycogen is stored in two main places in the body, the liver and inside the muscle fibers. And so glucose can then be released by the liver in order to produce enough glucose necessary to sort of get the muscle cells back going again. So again, this oxygen debt can be a problem. And so what happens is after exercise, the muscle fibers still require a lot of oxygen. They still require the production of ATP. And so what happens is, even when you're done, the body still continues heavy breathing in order to get that oxygen. And I might suggest to you, if you're vigorously exercising, a cool down period, because a cool down period is important in terms of getting in the body sort of tapering and still allowing the oxygen to build back up inside each of the muscle cells. And so now our conversation is going to emphasize the various muscle fiber types, and all of our previous discussion in this video has led us to this sort of peak right here. Is what I really, again, the title of the video is Types of Muscle Fibers, but in order to talk about that, we needed to have discussed everything that we have so far. And so there's three main muscle fiber types, but the truth is two of them are the most common. They're known as fast fibers or type 2, which are, when you name a fiber type 1 and type 2, it's not the most descriptive. I apologize for that. Wasn't me. But fast fiber is type 2 and slow fibers are type 1. And sometimes they're known as fast twitch, slow twitch. And then you have sort of intermediate fibers, and this is type 2A, intermediate fibers. So let's kick this around a little bit. So let's talk about fast fibers or type 2, fast fibers. So as the name implies, these particular muscle fibers can contract very quickly. They have large diameters. So the diameter of the muscle fibers is greater than in slow fibers. There's a large amount of glycogen reserve inside these cells and very few mitochondria. Now notice here I put this in bold, few mitochondria. In my judgment, when you're trying to remember all the different characteristics that differentiate between muscle fibers, it all comes down to the understanding that if this fast fiber has very few mitochondria, if you can just remember that, everything else kind of flows from it. Because as we'll see, this speaks to everything else in the conversation. And I want to also add that fast fibers have very strong contractions. As you can see here, these are the fast fibers, these ones that are sort of light in coloration, and they fatigue quickly. All right, these are some characteristics. Now the slow fibers, or type 1, contract slowly, but they're slow to fatigue, meaning that they're good for endurance. And so the fast ones are for short, quick bursts of powerful energy, and these are the ones that are more for long durations and endurance and slow to fatigue. But they're smaller in fiber and they're darker, and they have a high concentration of oxygen. And there's a variety of reasons for that. And there's a lot of capillaries that surround the slow fiber type. And also there is, in addition to hemoglobin, which you might be aware of is a protein that binds oxygen, there's another binding pigment called myoglobin, which is found inside the muscle cell, which also binds to oxygen. And as you may realize, when oxygen binds to these heme groups, it takes on a reddish hue. And so the fiber itself is darker in coloration and even dark red, if you will, as a result of myoglobin inside the slow fibers. And so when you can see here, there's a diagram, and I want to start off up here under the light microscope. This is sort of the white or fast fibers, again, lacking myoglobin, which gives it a whiter appearance. And here's red, So it's a darker coloration. Here's the capillary up here, and these little dark structures are the mitochondria right here, although they're very small. You might also notice that the fast fibers have a wider diameter, and the slow fibers have a less of a diameter. And so here's an example of a muscle in the eye and a muscle in our calf as an example uh, of a slow fiber and a fast fiber. Now, I'm going to hang out on this diagram for a little bit, or this table, I should say, because it's very important. Now, you can take a look over here at the intermediate, but I won't bring it up. I'm just going to contrast the type 2 and the type 1. In other words, the fast and the slow, and go down this with you. So cross section I mentioned before is large, so very powerful are these fast fibers, and small diameter. So again, fast means rapid peak to tension and a prolonged peak to tension. And again, the reason for this is that the slow fibers are going to need a lot of ATP production, and takes a little bit longer for fatty acids and glucose to be metabolized in the Krebs cycle and the electron transport system to produce the ATP necessary for prolonged endurance, and also high fatigue resistance. In other words, these do not tire as often. So it allows us to sort of stand around or walk around for an entire day. Okay. Now, these are red because the slow twitch are because they contain the myoglobin, and these are white. And low myoglobin content, high myoglobin content, lots of capillaries, scarce capillaries, and then many mitochondria. And so the many mitochondria I was mentioning before is the key to all of this. When you have many mitochondria, that means that you're able to metabolize lipids and carbohydrates aerobically to generate the maximum amount of ATP, and therefore you don't fatigue, and therefore you're able to endure. Whereas if you're a fast fiber and you don't have mitochondria, you're relying mostly on anaerobic metabolism, glycolysis. You have very few mitochondria, and you're basically just burning glucose at that point, which is perfectly fine. So our body kind of has a blend of these things, but I just want to point this out. If you're ever wondering why, when you're looking at chicken breast, which is sometimes referred as white meat, again, these are the fast fibers. They don't have a lot of myoglobin, and so they appear kind of lighter in coloration. And as it turns out, chickens don't really fly around too much. They're kind of they can fly, but it's short duration, and it's kind of powerful, if you will. But then they're back down, and what they mainly do is walk around. And so the thigh and the legs of chickens are red. So these are the ones that endure. These are the ones that are darker in coloration because there's more myoglobin. There's more mitochondria. There's more energy production. They don't fatigue, etc. Okay, and so again, humans, I mentioned this before, is kind of a mixture of these fibers, which give us a little bit of a pinkness, if you will, if you mix these together. Now, I also wanted to mention this conversation about, since we're talking about muscle fibers and types and stuff. So if you're talking about building muscle, enlarging muscle, it's referred to as hypertrophy. So muscle growth from heavy training. What that actually culminates in is the diameter of each muscle fiber will increase. There's more myofibrils that are being produced inside each of the cells. And there's also an increase of mitochondria and glycogen reserve. So all of this is very good. And I might also add is that when you have an increase in muscle, what's happening is that it increases your resting metabolic rate, which means that even when you're not exercising, your body is burning calories, which is often very good. Now, kind of as opposed to hypertrophy, what we have is atrophy. You may be familiar with this. It's the lack of muscle activity, and sometimes it's not intentional that we're not exercising. Sometimes, for example, if there's a fracture in our lower leg, we may have been placed in a cast, and so that these muscles haven't been working. And do you notice how this leg over here is reduced, the muscle size is actually reduced, and the tone and power is lost as well. And so this will eventually come back once atrophy can be recovered. Now, muscle tone, I've been referring to this, is when your muscles are not consciously like moving around, when you're sort of at rest, your muscles are still moderately contracting without motion. Yeah, I don't know if you knew this. It maintains posture and it maintains the firmness of the muscle. And again, the strength of the muscle is important because in muscle tone, when you can increase this and you have firm muscle or strong muscle tone, it increases the energy that's being used even at rest. And so this is a very important concept, muscle tone. Now, physical conditioning is how we can improve power and endurance. And so anaerobic, as I was mentioning before, in other words, without oxygen, sprinting, lifting weights very quickly are going to be using these fast fibers because they're very strong, but they fatigue quickly with strenuous activity. And so, sure, powerful, but you can't maintain it. Not good for endurance. But the more that you condition, in other words, the more you do exercise, you can improve. And so you can actually cause hypertrophy to occur, and therefore the muscles become more efficient. Now, you can also improve power and endurance aerobically, so prolonged activity, in other words, like a bike ride or a slow run. Now, this is supported by mitochondria, so it requires oxygen and it requires your nutrients, and that you can improve your endurance as well. So you can improve strength and you can improve endurance. And by training those fast fibers to be more like intermediate fibers. So what's going to happen is you're going to increase the number of mitochondria, and there's going to be a little bit more myoglobin produced, and the cells are going to not fatigue as much the more that you can do aerobic activity. And so, again, I'm not an exercise physiologist per se, but I think what I'm trying to get at, the punchline is a blend of different types of exercise are most important. So I would mix in like a medley of aerobic, cardiovascular, endurance types of exercise, as well as short burst sprints and power in lifting and weights. All of these things combined. The importance of exercise, wow. I mean, I can't say enough about it. Without it, I'll say that your muscles become flaccid or fatigued or atrophy, even just in a few days. And the muscle fibers begin to break down and the proteins become weaker and smaller over time. So exercise is really going to provide so much goodness. And so what I'll say about exercise is if it's important, and it is, you'll prioritize it. You'll not only give yourself the benefit of exercising, but it'll give you energy. And I would also add, I would choose something that you enjoy doing so that you'll want to do it more. And again, this is maybe obvious too, but you might want to exercise with other people so that it's social. And so you can, other people can encourage you, you can encourage other people, and it becomes a routine and something that you look forward to, not something that you dread. Okay, so I hope you enjoyed that video on muscle fiber types. Thanks for watching.